So let's now we talk in detail about the metabolism of cholesterol. Here we will talk about the synthesis of cholesterol. Cholesterol is synthesized almost in all the tissues, but the major organs or major tissues in which it is synthesized as liver and intestine, and also in the placenta and kidneys, and of course intestines also. So almost in all the tissues it is produced, but the major production organs or tissues are the liver and the kidneys and intestine. There are many enzymes. You will see that it's a very complex process, the synthesis of the cholesterol. There are many enzymes which are involved in its synthesis. These enzymes are present in the cytosol. They require the membranes of the smooth endoplasmic reticulum. They also require the enzymes of the peroxisomes. The cholesterol synthesis is also very well regulated. One of the way to regulate it, and we will see it also, that is through assessing the concentration of the cholesterol in the body. And there are some other regulatory pathways also, regulatory mechanisms which balance the synthesis and excretion of cholesterol. So as with all the or most of the other lipids, the cholesterol synthesis also start with the acetyl-CoA. Here you can see two acetyl-CoA molecules. Under the action of thiolase, they form a 4-carbon compound, the acetoacetyl-CoA. Keep in mind, when we are talking about synthesis, you should focus on the number of carbon atoms also, not only on the reactant and products, or not only on the enzymes which are involved. Also, you should focus on how the number of carbon atom is changing from one compound to the other, or from one product to the other. So the 4-carbon acetyl-CoA is converted by the action of HMG-CoA synthase enzyme, and we get the HMG-CoA. As a result of this, 3-hydroxy-3-methyl-glutyl-CoA. This 3-hydroxy-3-methyl-glutyl-CoA, I have told you about this, I have already read it. So 3-hydroxy-3-methyl-CoA is a very important enzyme because it is acted upon by different compounds or different factors which regulate the synthesis of cholesterol. So as with the other lipids also, where the starting material is acetyl-CoA, here again we have two acetyl-CoA as the starting material, which combine with each other to give rise to acetoacetyl-CoA. Acetyl-CoA is here is 2-carbon compound. Of course, you get in the product 4-carbon compound, and then from here you get the HMG-CoA 3-hydroxy-3-methyl-glutyl-CoA, which is a 6-carbon compound, and the enzymes involved is HMG-CoA synthase. So keep in mind here that is a complex process, the synthesis of cholesterol. Many products are formed. Some of the things you have to keep in mind, of course, is the enzymes which are involved, of course, bacterial products which are involved, but also very important is that how the carbon atom number is changing. For instance, here we started with 2-carbon compound, there comes 4-carbon compound, and finally we have 6-carbon compound. So this is also you should keep in mind. Then a very important reaction, perhaps as far as the regulation of cholesterol is concerned, this is the most important reaction, that is the conversion of HMG-CoA into mevalonate. So in this reaction, this is a reduction reaction in which NADPH is utilized, and The enzyme is HMG-CoA reductase. So this is a rate limiting reaction, that is it is regulated. It regulates the synthesis of the cholesterol, and this enzyme is acted upon by many factors which eventually regulate the synthesis of cholesterol. Now you have seen that we started with, for instance, 2 carbon compound, and until the very rate limiting step, when you get the mevalonate, you get the 6 carbon compound. The next step from the 6 carbon compound onward until we get 30 carbon cholesterol compound, the reactions are quite complex and very energy intensive, and I will need your real focus in understanding those reactions.